Imagine being diagnosed with a degenerative disease, and current treatments are limited. Maybe they're only managing symptoms. But what if your doctor told you there was a chance to actually repair the damage using your own cells? That's the potential of what we're diving into today.
Yeah.
Induced pluripotent stem cells, or iPSCs. It's a bit of a mouthful, but trust me, the science is mind-boggling.
It really is. iPSCs are like unlocking a cellular time machine. We can take adult cells, like skin cells,
Yeah.
and rewind them back to a state where they can become any cell in the body.
Okay, so rewind my skin cells back to when exactly?
Think embryonic-like. They're not actually embryos, but they have that same potential to become anything. Heart cells, neurons, you name it. It's a total game changer for medicine.
So how did scientists even discover they could do this? Was it an accident or years of dedicated research?
A bit of both, actually. Back in 2006, a scientist named Shinya Yamanaka was determined to crack the code of cellular reprogramming. He was convinced that we could reverse the clock on adult cells. It took years of painstaking research, but he finally discovered that by introducing just 4 specific genes, think of them like molecular switches, he could flip adult cells back to this pluripotent state.
Hold on, so just 4 genes can completely reprogram cells' destiny. It seems too simple.
It's simple in concept, but incredibly complex in execution. These genes, called transcription factors, orchestrate a cascade of changes within the cell, essentially erasing its memory of being a skin cell and giving it the potential to become anything. It was a Nobel Prize-worthy discovery for a reason.
A Nobel Prize. Okay, I'm really intrigued. So let's talk applications. We got these cellular blank slates. What can we do with them?
Well, one of the most exciting areas is personalized medicine. Imagine being able to study a disease in a dish, using cells that are genetically identical to yours.
Wait, you're saying they can take my skin cells, turn them into iPSCs, and then use those to study, say, a heart condition I might have?
Exactly. Let's say you have a family history of heart disease. Researchers could take your skin cells, reprogram them into iPSCs, and then guide those iPSCs into becoming heart cells. Now they have a dish of heart cells with your unique genetic makeup, allowing them to study how heart disease might develop in your case specifically.
Wow. That's incredibly powerful. It's like having a personalized window into your own potential health risks. But studying disease is one thing. Can we use iPSCs to actually treat diseases?
Absolutely. That's where regenerative medicine comes in. Take Parkinson's disease, for example. It's caused by the loss of specific brain cells that produce dopamine. Scientists are working on using iPSCs to generate those dopamine-producing cells and then transplanting them into patients' brains to replace the lost cells.
So you're essentially replenishing the brain with healthy cells that are a perfect match for the patient, minimizing the risk of rejection. It's like hitting the reset button on the disease.
That's the goal. And it's not just limited to Parkinson's. Scientists are exploring iPSC-based therapies for a wide range of conditions, from spinal cord injuries to macular degeneration.
This is incredible, but wouldn't growing entire organs be even more impactful? I've heard about scientists creating mini organs from iPSCs. How close are we to actually transplanting those?
You're right. Lab-grown organs would be a game changer. Scientists have already created things like mini livers and mini kidneys, but these are still in the early stages. One of the biggest challenges is getting these mini organs to grow large and complex enough to function like their real-life counterparts. And we also need to figure out how to integrate them seamlessly into the body with a proper blood supply and nerve connections.
So it's a long road ahead. But it sounds like we're making progress. Beyond regenerative medicine, what other areas are benefiting from iPSCs?
Drug discovery is another major area. Traditionally, testing new drugs has relied heavily on animal models, but what works in a mouse doesn't always translate to humans.
Right, we're not just giant mice. Different species, different biology, different reactions to drugs.
Exactly. With iPSCs, we can create human cells and tissues to test drugs on, which is a much more accurate and relevant model. Plus, using iPSCs derived from patients with specific diseases allows us to test drugs that are tailored to those diseases, leading to more effective treatments with fewer side effects.
So iPSCs are essentially speeding up the drug discovery process and leading to safer, more effective treatments. It's a win-win situation.
It is, and the pharmaceutical industry is already taking notice. Several drugs that have been developed using iPSC-based testing are now in clinical trials, and many more are on the horizon.
This is seriously impressive. So we've talked about personalized medicine, regenerative therapies, and drug discovery. What other surprises does this technology have in store?
Well, iPSCs are also helping us understand the aging process itself. By comparing iPSCs from young and old individuals, scientists are starting to unravel the molecular mechanisms of aging and identify potential targets for therapies that could slow down or even reverse age-related decline.
Okay, now you're just teasing me with the fountain of youth. But seriously, the potential of iPSCs to tackle age-related diseases is incredibly exciting.
It is. Think about conditions like Alzheimer's, heart disease, and osteoarthritis. These are all linked to the aging process. And if we can understand aging at a cellular level, we might be able to develop new treatments to prevent or delay these conditions.
So iPSCs are not just about treating diseases, they're about understanding the fundamental processes of life and potentially extending our healthy lifespan. That's pretty mind-blowing.
It really is, and the field is still in its infancy. We've only scratched the surface of what iPSCs can do.
It's amazing to think that these tiny cells hold so much promise for the future of medicine, but with such a powerful technology, there are bound to be some challenges, right?
Of course. There are still technical hurdles to overcome, ethical considerations to address, and the need to ensure equitable access to these potentially life-changing therapies. But the progress being made is incredibly encouraging.
It sounds like we've only just begun to explore the vast potential of iPSCs. This is definitely a topic we'll need to revisit in a future deep dive. But before we move on, let's shift gears for a moment and delve into some of the challenges and ethical considerations surrounding iPSC technology. I'm curious to know, what are some of the biggest concerns that scientists and ethicists are grappling with?
One of the biggest concerns is the risk of tumor formation.
Okay, tumor formation. That sounds a bit scary. How does that happen?
Well, remember how iPSCs have this remarkable ability to differentiate into any cell type. Sometimes that can go awry. If the reprogramming process isn't perfectly controlled, the cells can start dividing uncontrollably, potentially leading to the formation of tumors.
So safety is a major consideration. Are there ways to minimize this risk?
Absolutely. Scientists are working on several fronts. They're refining the reprogramming methods, developing stricter quality control measures for iPSCs, and exploring new ways to monitor the cells after transplantation to ensure they're behaving as expected.
That's reassuring. What are some other challenges researchers are trying to solve?
Another hurdle is the efficiency of the reprogramming process. Turning adult cells into iPSCs isn't always a smooth ride. It can be time-consuming, expensive, and the success rate isn't always what we'd like it to be.
So improving the efficiency and reliability of the process is key to making iPSC-based therapies more accessible.
Exactly. And there's a lot of research focused on finding ways to make reprogramming faster, cheaper, and more consistent. Scientists are experimenting with different combinations of genes, using new delivery methods, and even exploring the use of small molecules to boost the reprogramming process.
It sounds like scientists are pulling out all the stops to optimize this technology.
Wow.
What about the ethical considerations? What are some of the big questions that are being debated?
One of the most complex issues is the potential for germline modification.
Germline modification. Refresh my memory on what that means.
It means making genetic changes to iPSCs that could be passed down to future generations. Imagine, for example, using iPSCs to correct a genetic defect in an embryo. That correction would not only benefit the individual, but also their children and grandchildren.
I can see how that raises a lot of ethical questions. It's like we're on the verge of editing the very blueprint of humanity.
Exactly. It's a powerful tool with far-reaching consequences. We need to have careful and open discussions about where to draw the line, what types of genetic modifications are acceptable, and what are the potential risks and benefits for individuals and society as a whole.
These are weighty questions that we can't shy away from. But beyond the ethical considerations, there's also the issue of access and equity. Right? We need to ensure that everyone benefits from these advances, not just a privileged few.
You're absolutely right. iPSC technology has the potential to transform healthcare, but we need to ensure that those benefits are accessible to everyone, regardless of their socioeconomic status or geographic location.
So we need to address the cost of these therapies, ensure that they're available in different parts of the world, and make sure that healthcare systems are equipped to deliver these new treatments effectively.
Precisely. It's not just about the science. It's about building a healthcare system that is equitable and sustainable so that everyone can benefit from the promise of iPSCs.
It's a complex landscape with both incredible opportunities and significant challenges. And as we move forward with iPSC research, it's crucial that we consider the ethical and economic implications of this powerful technology. But let's not lose sight of the incredible progress that's been made. We've come a long way since the discovery of iPSCs in 2006. And the future of medicine is looking brighter than ever. Stay tuned for part 2 of our deep dive, where we'll explore some of the cutting-edge advancements in iPSC research and delve deeper into the potential of this technology to revolutionize how we treat and prevent diseases. Welcome back to our deep dive into the world of induced pluripotent stem cells.
It's remarkable how these tiny cells are opening up entirely new avenues in medicine, isn't it?
Absolutely. In the last part, we talked about the potential of iPSCs for personalized medicine, regenerative therapies, and even drug discovery. But we also touched on some of the challenges, like tumor formation and the ethical considerations around germline modification. Now, I'm curious to know more about the cutting edge of this field. What are some of the most exciting advancements happening right now in iPSC research?
Well, one area that's really exploding is the use of iPSCs to create disease models that are far more accurate and sophisticated than anything we've had before.
You mean going beyond just having a dish of heart cells or neurons?
Exactly. Scientists are now creating 3D structures called organoids, which are essentially miniature versions of organs grown from iPSCs. These organoids mimic the complexity of real organs much more closely, with different cell types interacting in 3D space just like they do in the body.
So instead of just studying individual cells, we can now study how cells behave and interact within a more realistic organ-like environment.
Precisely. And this is a game changer for understanding complex diseases that involve multiple cell types and organs. For example, researchers are now using iPSC-derived organoids to study conditions like cystic fibrosis, which affects the lungs, pancreas, and other organs. These organoids allow scientists to observe how the disease progresses, test new drugs, and even personalize treatments based on a patient's specific genetic makeup.
That's incredible. It's like having a personalized mini lab for each patient. Are there any other areas where organoids are making a big impact?
Absolutely. Brain organoids, sometimes called mini-brains, are revolutionizing our understanding of neurological disorders. Researchers are using them to study conditions like Alzheimer's disease, Parkinson's disease, and autism spectrum disorders. These mini-brains can't think or feel, of course, but they do exhibit some of the complex structural and functional features of the human brain, allowing scientists to observe how these disorders develop and test potential treatments in a way that was never before possible.
It's mind-boggling to think that we can now grow miniature versions of brains in a dish. It seems like science fiction, but it's happening right now. So we've got these incredible disease models. How are iPSCs changing the way we develop new drugs?
Well, as we discussed earlier, iPSCs are allowing us to move away from animal models and test drugs on human cells, which is much more accurate and relevant. But beyond that, iPSCs are also being used to develop entirely new types of drugs based on a deeper understanding of the disease process itself.
Okay, give me an example. How are iPSCs leading to these new drug discoveries?
Let's take spinal muscular atrophy, or SMA, a devastating genetic disease that affects motor neurons and leads to muscle weakness and paralysis. Using iPSCs, scientists were able to identify a specific protein that plays a key role in the disease process. They then developed a drug that targets this protein. And in clinical trials, this drug has shown remarkable results, dramatically improving the lives of children with SMA.
That's an incredible story of how iPSCs can translate into real-world treatments and hope for patients. Are there any other examples of iPSC-based drugs that are showing promise?
Absolutely. There are several drugs in development for conditions like heart disease, diabetes, and even cancer that are based on insights gained from iPSC research. And one area that's particularly exciting is the development of personalized cell therapies using iPSCs.
Personalized cell therapies. Remind me how those work again.
Essentially, it involves taking a patient's own cells, reprogramming them into iPSCs, and then differentiating those iPSCs into the specific cell type that the patient needs. For example, if a patient has type 1 diabetes, which is caused by the destruction of insulin-producing cells in the pancreas, we could potentially use iPSCs to generate new insulin-producing cells and transplant them back into the patient.
So you're essentially replacing the damaged cells with healthy cells that are a perfect genetic match, eliminating the risk of rejection. That's an incredible concept. Are there any personalized cell therapies currently available?
Yes, actually. There's a form of personalized cell therapy for macular degeneration, a leading cause of vision loss. That uses iPSC-derived retinal pigment epithelial cells. These cells are transplanted into the eye, where they can help to preserve and even restore vision.
That's a remarkable example of how iPSCs are already making a difference in patients' lives. What are some other areas where personalized cell therapies are being explored?
Oh, the possibilities are vast. Scientists are investigating iPSC-based therapies for a wide range of conditions, from Parkinson's disease and spinal cord injuries to heart failure and liver disease. And one of the most exciting frontiers is the development of iPSC-derived immune cells that can be engineered to attack cancer cells.
So you're talking about creating personalized cancer-fighting cells that are tailored to each patient's specific tumor.
Exactly. This approach, called CAR T-cell therapy, is already showing remarkable success in treating certain types of leukemia and lymphoma. and researchers are working to expand its use to other types of cancer.
It's incredible to think that we might one day have personalized immune cells that can hunt down and destroy cancer cells with pinpoint accuracy. This is truly revolutionary stuff. But I imagine there are still challenges in bringing these therapies to a wider patient population.
You're right. One of the biggest challenges is cost. Developing and manufacturing personalized cell therapies is currently very expensive.
So accessibility and affordability are key hurdles that need to be addressed.
Absolutely. We need to find ways to make these therapies more accessible to everyone who needs them, not just those who can afford them. Researchers are working on developing more efficient and cost-effective methods for producing iPSCs and differentiating them into the desired cell types. And we also need to ensure that healthcare systems are equipped to deliver these complex and personalized treatments effectively.
It's clear that iPSCs are not just about the science. It's about the people. It's about ensuring that everyone can benefit from these advances, regardless of their socioeconomic status or geographic location. But beyond the practical considerations, there are also ethical questions that we need to keep in mind as this technology continues to evolve.
You're absolutely right. As with any powerful technology, we need to be mindful of the potential risks and unintended consequences. We've already touched on the concern about germline modification, but there are other ethical considerations as well.
Such as?
Well, one issue is the potential for iPSCs to be used for enhancement purposes, rather than strictly for treating diseases.
You mean like using iPSCs to make people stronger, smarter, or more attractive?
Exactly. As the technology advances, we may be able to use iPSCs to enhance physical or cognitive abilities, but this raises a whole host of ethical questions. Should we be using this technology to create superhumans? What are the implications for social justice and equality if only certain people have access to these enhancements? These are complex and nuanced questions that we need to grapple with as a society.
It's a lot to consider, but it's crucial that we have these conversations now. Before the technology outpaces our ethical understanding, we need to establish clear guidelines and regulations to ensure that iPSC technology is used responsibly and ethically.
I completely agree. The power of iPSCs is undeniable. But with that power comes responsibility. We need to ensure that this technology is used to improve human health and well-being, not to exacerbate existing inequalities or create new ones.
It's a delicate balance, but it's one we must strive for. And as we continue to explore the vast potential of iPSCs, it's essential that we keep these ethical considerations at the forefront of our minds. But let's not lose sight of the incredible progress that's been made. From disease modeling to drug discovery and personalized cell therapies, iPSCs are transforming the landscape of medicine. And the future holds even more promise. Stay tuned for the final part of our deep dive, where we'll delve deeper into the potential of iPSCs to revolutionize how we understand and treat diseases, and explore what this technology might mean for the future of healthcare. Welcome back. We've been on quite a journey through the world of induced pluripotent stem cells, haven't we?
It's been fascinating to explore how these versatile cells are reshaping our understanding of biology and the possibilities for medicine.
Absolutely. We've talked about how iPSCs are revolutionizing disease modeling, drug discovery, and even personalized therapies. We've also touched on some of the challenges and ethical considerations. And I'm curious, what do you think the future holds for iPSCs? What's on the horizon for this technology?
Well, I think we can expect to see even more incredible breakthroughs in all areas of iPSC research. For instance, in disease modeling, we're moving towards creating even more sophisticated and personalized models, like those 3D organoids we discussed. Imagine using your own cells to create a mini model of your heart or your brain, allowing doctors to study how diseases might affect you specifically and tailor treatments to your unique needs.
It's almost like having a personalized avatar for your health. But beyond disease modeling, what about actual treatments? How close are we to seeing widespread use of iPSC-based therapies?
We're getting closer every day. The field of regenerative medicine is advancing rapidly, with clinical trials underway for a growing number of iPSC-based therapies. As we discussed earlier, there's already a personalized cell therapy available for macular degeneration. And promising results are emerging for conditions like spinal muscular atrophy, Parkinson's disease, and type 1 diabetes.
So it's not just a theoretical possibility anymore. iPSCs are already making a real difference in people's lives.
Exactly. And as the technology matures and becomes more efficient, we can expect to see a surge in iPSC-based therapies becoming available. But it's not just about the number of therapies, it's also about refining those therapies, making them even safer, more effective, and more accessible.
What are some of the key areas where researchers are focusing their efforts to improve iPSC-based therapies?
Well, one major focus is on improving the safety and efficiency of the reprogramming process. Scientists are constantly refining the methods used to turn adult cells into iPSCs, making the process faster, cheaper, and less prone to errors. They're also working on developing better ways to control the differentiation of iPSCs, ensuring that they reliably turn into the desired cell type without any unwanted side effects.
So it's about fine-tuning the cellular machinery, so to speak. But what about the cost of these therapies? How can we make sure they're accessible to everyone who needs them?
That's a critical issue. Right now, many iPSC-based therapies are prohibitively expensive, but there's a lot of effort going into developing more cost-effective methods for producing iPSCs and manufacturing these therapies.
So making it more like mass production instead of a bespoke one-off process for each patient.
Exactly. And there are some promising avenues being explored, like using bioreactors to grow large quantities of iPSCs and developing off-the-shelf cell therapies that can be used for multiple patients, rather than creating a unique therapy for each individual.
It sounds like there's a real push to make these therapies more widely available. But beyond the practical aspects, there are also the ethical considerations that we need to keep in mind.
You're absolutely right. As this technology becomes more powerful and more readily available, we need to be prepared for the ethical challenges that will inevitably arise.
What are some of the key ethical questions that you think we need to be thinking about?
Well, we've already talked about the potential for iPSCs to be used for enhancement purposes rather than just treating diseases. That's a complex issue with no easy answers. But there are other concerns as well. For example, who should have access to these therapies? How do we ensure that they're distributed fairly regardless of someone's socioeconomic status or geographic location? And how do we prevent the creation of a genetic elite where only the wealthy can afford to enhance their abilities or those of their children?
These are weighty questions.
Yeah.
And it's clear that we need to have open and honest conversations about the ethical implications of iPSC technology as it continues to evolve. But I think it's also important to remember the incredible potential of this technology to alleviate suffering and improve human health.
Absolutely. We've seen how iPSCs are already giving hope to patients with devastating diseases, and I truly believe that this is just the beginning. As we continue to unravel the mysteries of these remarkable cells, I'm confident that we'll discover even more ways to use them to benefit humanity.
It's inspiring to think about the possibilities. And as we wrap up our deep dive, I want to thank you for sharing your expertise and insights. It's been an incredible journey through the world of iPSCs.
It's been my pleasure, and I hope this deep dive has inspired you to learn more about this groundbreaking technology and its potential to transform the future of medicine.
I know it certainly has for me. And for our listeners out there, we encourage you to keep exploring this fascinating field. The future of medicine is being rewritten one cell at a time, and it's a story you won't want to miss. Thanks for joining us on the deep dive. Until next time, keep learning,